Hi, good morning. Uh, so before introducing to Katie Solari for her presentation, I will like to introduce myself. My name is Shakil Ahmed, and I have PhD in Wildlife Ecology from Quaid-e-Azam University, Pakistan. My doctoral research focused on understanding snow leopard population dynamics and relation to resource availability using non-invasive genetic approaches and spatial capture-recapture models. My work emphasizes the application of molecular ecology and advanced spatial and statistical analysis to estimate snow leopard population size, density, and prey availability of the snow leopard. My broader research interests include large carnivores and their prey ecology, human-wildlife interactions, and the use of non-invasive tools such as camera trapping and non-invasive genetics to support evidence-based wildlife conservation and management, particularly in northern Pakistan. Now, I would like to introduce Catherine Solari for her presentation. Before her presentation, Dr. Katie Solari has a PhD in wildlife and evolutionary biology from Stanford University. Uh, Catherine is currently a research scientist in the Petrie Lab at Stanford University and the Associate Director of the Program for Conservation Genomics. Katie works focus on the developing methods and generating genetic data that can be used to aid conservation management. This includes producing genomic dataset for species of conservation concern and distilling this data on down to the SNP panel that can be used to gain meaningful information from non-invasive. Now I would like to uh, uh, now I would like to call Katie Solari to present her genomic works. That is the basic question of the her work is how genomics can inform conservations. So now, up to you, Katie.
Awesome. Thanks so much, Shaquille. I appreciate the introduction. Yeah, so I'm Katie Solari. I'm currently at Stanford University and part of the program for conservation genomics. And yeah, I'm really excited to tell you guys today about some of the work we've been doing on snow leopards, using genomics to better understand their species history, their current distributions, and then to design these non-invasive monitoring tools for their long-term conservation. I wanted to start by saying this photo is of Raj, a snow leopard from the San Francisco Zoo. And the snow leopards at the San Francisco Zoo are the only ones I've ever seen. So I haven't been lucky enough to do any field work for snow leopards. So all of the work that I'll talk about has been through extensive collaborations. But I have been lucky enough to study one of the prey items of snow leopards. I did my PhD work on pikas, and this work brought me to snow leopard habitat. So I have spent quite a bit of time in the Himalayas of India and Nepal. This is in Himachal Pradesh. So I've seen snow leopard tracks and stuff, but anyway, just to kind of put it out there that I'm not a snow leopard field biologist. And yeah, and I'll be highlighting my collaborations as I go through the talk. So to start off with a bit about the program for conservation genomics. Sorry, I'm trying to place this so I don't get distracted by everything. So the program for conservation genomics our main goal is to capitalize on genomic technology to develop methods to monitor species non-invasively. And so the methods that I'm going to be talking to you about today can be applied to any species of conservation concern. So any of the species shown here, it's very versatile. And the main thing that we're trying to do is to capitalize on the plummeting cost of DNA sequencing information, which has just become affordable to the generally underfunded conservation sector in the last few years. So here I'm showing the cost per 3 megabases of DNA sequence. And the reason that I have three gigabases, the reason I have three gigabases here is that's the average length of a mammal genome. So this is the cost to read through one time, like an average mammal genome. And in 2001, this cost $30 billion. And in 2022, this costs $30. And today, in 2026, we're paying closer to this $3 mark. And so that's for 3 gigabases of DNA, which is 3 billion base pairs of DNA information. So this precipitous drop has really has really made this type of information accessible to the conservation sector. And the movement of this data to the conservation sector has just been really slow. And so this is where we're kind of trying to step in and speed up this movement of information into this different space. So, uh, so this generally, conservation programs that are using genomic data are using invasive sampling. So this is going to be blood or tissue samples. This is the type of sample that gives you the best data. And so, as those of you who have done work on snow leopards in the field, I'm sure you can understand how hard it is to capture a snow leopard to get a blood or tissue sample. So this is extremely invasive, expensive, and time consuming. Even for work that we do on much more common species, such as waterbuck, which are in number in the tens of thousands, where we study them in Africa. This is still a very elaborate process to get blood or tissue samples from them. It involves helicopter work that is very stressful for the whole ecosystem and very expensive. So, invasive sampling has lots of drawbacks. And so the key thing that we're focusing on is being able to move away from invasive sampling and more towards non-invasive sampling. So being able to get very meaningful, insightful information from fecal samples. And the benefit here is here is Shaquille with all of his samples that he brought to our lab from Pakistan, that you can just collect a huge amount of samples in the same amount of time that you might get one tissue sample. And again, the same is true from even more numerous samples here, where this is a colleague of mine laying out gloves to highlight all of the fresh fecal samples that I'm about to go collect. So even for more numerous samples, you can just do a sweep and collect dozens of samples, whereas it would be much more time consuming and expensive to collect one tissue sample. So there's lots of differences though between invasive sampling and non-invasive sampling. As I kind of alluded to, with invasive sampling, blood or tissue samples, you can really get all the information you would ever want from a sample. You can get the whole genome. So you can it's reasonable to assume that you're going to be able to sequence all 3 billion base pairs of that animal's genome. Whereas with non-invasive sampling, so this is a snow leopard sample, a snow leopard fecal sample that I found in the field when I was doing pico work. And my PSA is don't don't hold a fecal sample without a glove. So do as I say, not as I do. This I wasn't actually doing snow leopard work at the time, but if you do want to study the feces of an animal, do wear a glove because I'm contaminating this snow leopard feces with my own DNA in this photo. Um, but from this fecal sample, there's only kind of residue amount of snow leopard DNA. And so we can't really get all 3 billion base pairs of DNA. What is more reasonable to aim to get is like between 100 and 1,000 base pairs of DNA. And specifically here, I'll start kind of dropping in some lingo that not all base pairs of DNA are informative. So here, like this TT, this is these are like two genetic sequences, let's say from two different snow leopards. And both of them has to have T at this base pair. So that's non-variable and not super informative. Whereas this base pair, there's one A and one G, and this is variation. And this is what we would call a single nucleotide polymorphism or a SNP. And this is informative because it differentiates these two individuals. So what we can hope to get from a fecal sample like this is like 100 to 1,000 SNPs. And so those are kind of the two methods that we employ are whole genome sequencing from invasive blood or tissue samples. You sequence the whole genome, but then from fecal samples, you can employ a SNP panel where you just pull out these 100 to 1,000 SNPs that are going to be informative and allow you to get meaningful information from this marginal sample. So I'm going to run through the steps of how we go about developing a SNP panel for as our non-invasive monitoring tool. So the first thing that you need is to have a high quality reference genome for the species of interest. So in this case, that would be a snow leopard. The second thing that you need is whole genome sequencing data for individuals from across the species range. And again, as I just said, the whole genome sequencing is best from blood or tissue samples. So this involves getting these invasive samples. And with this data, we want to make what we kind of call a genoscape or a geographic distribution of the genetic diversity of the species. So this is a cartoon of the snow leopard range showing what this could look like. So if we sampled snow leopards from across their range, maybe we would find that this area up here in bright red is genetically distinct. And this area in orange is its own genetically distinct population, and this population down here in purple is genetically distinct. So the colors here are just designating genetically different subsections of their range. So that's what we would kind of hope to understand from doing this whole genome sequencing across the range of the snow leopard. And then once we have that information, we can boil down that whole genome sequencing information to just identifying SNPs that are meaningful to the question that we have at hand. So the two main questions that I'll be talking about are individual ID and geographic assignment. And so individual ID would be mainly drawing SNPs that are variable within a population, whereas geographic assignment would be drawing on SNPs that are really diagnostic of different populations in the snow leopard range. So in the cartoon here, I'm showing that this orange population of snow leopard has maybe these two SNPs that are only found in that region, in that orange region. And so if we're able to sequence those two SNPs, we can then say that sample is from that orange region because those orange SNPs are only found in that orange region. So yeah, so that's kind of how we go about that part. And then once you have identified the SNPs that you want to target, you can develop the SNP panel and use that SNP panel on low quality samples. And low quality means low concentrations of snow leopard DNA. So that's going to be fecal samples, but it can also be hair, saliva, or confiscated materials. And then we're able to use that SNP panel to just extract those few meaningful pieces of information and answer the question that we started with. So now I'm going to jump into snow leopards and highlight how we've gone through this with snow leopards. So many of you probably, know more about snow leopards than I do, but going into this project, there were numerous knowledge gaps with snow leopards because, again, as many of you know, they're extremely elusive and hard to study. And so there's lots of things about their general biology that just aren't well characterized because they're so hard to observe. So that includes population size, both current and historic, connectivity across the landscape, as well as levels of genetic diversity. So these were some of the things that we had in mind to try and understand better. So in the case of the snow leopard, our group had already made a new high quality reference genome for snow leopard. So this first step was done. So we jumped in on number 2 here, which is creating whole genome sequencing data for individuals from across the range. And when we started this, there was only genomic data available for 3 snow leopard individuals. So 1 wild individual from Mongolia and 2 captive individuals. And if you look at how expansive their range is here, 3 individuals is not going to do a great job at telling us about the genetic diversity across the landscape for such a widespread species. So we started out by trying to accumulate blood and tissue samples to generate this whole genome sequencing from across the range and fill in these gaps. And so these samples largely came from blood samples from collaring efforts, as well as tissue samples from carcasses that different field biologists encountered. And as you can imagine, this was a hugely collaborative effort with all of the groups shown here involved. I've listed here all of the co-authors on this work too, just to highlight what a big effort this was. And this basically took many, many years of just compiling all of the blood and tissue samples that all of these groups had collected over the last 20 years. So this was kind of only possible by all of these groups coming together. And as a result of this effort, um, we we were able to accumulate whole genome sequencing data for 41 snow leopards. So this included 35 wild individuals from 7 different countries, which are highlighted here on the map, and then 6 captive individuals. So this increased the amount of genomic data available for snow leopards by an order of magnitude. I will highlight here, I'll come back to this, but you'll notice from the map here that we didn't have any samples from China or Nepal or Bhutan, like most of the Himalayas. So uh, we didn't capture the whole range, but I'll circle back as to how we're addressing this and what updates we have on this now. But still, this is way better than the 3 samples and 1 wild sample that we started with. And you'll see that we can still get a bunch of meaningful information from this data set. So the first thing that we do using this whole genome sequencing information is to call SNPs. So as I mentioned, the genome is about 3 billion base pairs long, and we identified among those 3 billion base pairs about 1.5 million sites where there was variation among these 41 snow leopards. So we took that 1.5 million SNPs, and that's what we did our genetic analyses with. So these variable sites across the genome. And the first thing that we did was to make this look at the data in this PCA. So this is a principal components analysis where each point is a different snow leopard sample, and points that fall closer together are more genetically similar. And this is using all of that 1.5 million SNPs. And you can see that there's generally 3 different groups here. So we see 3 different distinct snow leopard groups. And if we look at where these are from, they separate geographically. So we see that Russia and Mongolia is one distinct genetic group, and then Tajikistan, Afghanistan, and Pakistan are a second distinct genetic group. And then our samples from Kyrgyzstan in blue here group with our captive samples. So 2 things I want to note here. One is you might notice that we only had 1 sample from India, and this sample was quite genetically distinct. And because we only had 1 sample and it was off on its own, we couldn't really include it in these analyses because we couldn't say whether or not it was actually genetically distinct or it was just something random about the sample. So anyway, I can talk more about that after, but that's why that isn't included. And then the other interesting thing that we see here is that the captive samples, which are zoo samples from the U.S. and Europe zoo population, are falling really closely with the Kyrgyzstan group. And this is interesting because we actually have the pedigree information for all of these samples. So these samples are shown here and filled in black. And first of all, we can see that they're genetically related to each other. But up here we have the wild sources of that seeded this captive zoo population. And some of them are from known areas, but a lot of them are from unknown locations. And what this tells us is that a lot of these animals that came from the wild to seed the zoo population likely came from Kyrgyzstan or Kazakhstan or that general area, which is actually consistent with what our colleagues at the Snow Leopard Trust say they kind of were familiar with. So that's kind of an interesting insight, I think. Um, Perhaps our most interesting finding from this data set was when we looked at the observed heterozygosity. So here, each point is a individual. And here I have different Panthera species and subspecies. So these are different tiger subspecies, lion, jaguar, Asian leopard, and African leopard. And observed heterozygosity is basically a measure of how genetically distinct, the level of genetic diversity of that individual. So the number of sites across the genome where it's variable. And so basically what we see by looking at these other Panthera species, that African leopards are more genetically diverse than other Panthera subspecies, which was already known. This is all publicly available data. We added in other big cats, so pumas here as well as cheetahs. When we add cheetahs in, we see that cheetahs have a lower genetic diversity than these other big cat species, which was also known. People might be familiar that cheetahs are kind of known as like the poster child for low genetic diversity. And then the surprising part was when we added our snow leopard data to this, and we see that snow leopard, every single snow leopard sample that we have has a lower genetic diversity than any other big cat species. So this was a surprising finding that was previously kind of underappreciated how little genetic diversity snow leopards have. And so that kind of led to the question of like, why is this the case? Why is it that snow leopards are the least genetically diverse of all the big cat species? And there's two main ways that species can end up with really low genetic diversity. One is high levels of recent inbreeding. And this is what has resulted in cheetahs being having very low genetic diversity. So inbreeding generally occurs when a population goes through a bottleneck and there's only a few individuals left, and they kind of have no choice but to mate with each other, even if they're somewhat related. And then when you have relatives mating, their offspring have low genetic diversity because they're getting their DNA from their mom and their dad are similar because their mom and their dad are related. So this is why cheetahs have low genetic diversity. We looked at this. This is an inbreeding coefficient as a measure of inbreeding. And here, snow leopards in gold compared to other Panthera species, we can see that they're not significantly different in general. They have a very standard level of inbreeding compared to other big cats. So this tells us that this isn't what's causing it. If it was what was causing it in snow leopards, then we would expect snow leopards to display a much higher inbreeding coefficient compared to other species. So the other option is that you can end up with low genetic diversity if the population has always been small. And that's because you need a big population to generate new genetic diversity to end up with a species that has high genetic diversity. So we looked at this in snow leopards. This plot. The details aren't super important, but this is basically a plot that estimates the effective population size through the evolutionary history of the species. So on the x-axis is time, where we have 1,000 years ago on the left and then 1,000,000 years ago on the right. And the different lines here are different snow leopard populations. And basically, what we see here is that snow leopards likely never had more than 30,000 individuals, even at their peak. And through most of their evolutionary history, they likely only had on the order of 10,000, 15,000 individuals max. So, as opposed to other big cat species where there were potentially hundreds of thousands of individuals, snow leopards never got to that population size. So, this is consistent with why we're seeing low genetic diversity in snow leopards. They've just always kind of cruised along at a small population size, and so they've never amassed a ton of genetic diversity in the population. And this also makes sense if you think about their ecology. So snow leopards live in these really remote habitats where there isn't a lot of vegetation, there isn't a lot of animals for them to feed on. So these habitats that they've evolved in and specialized in can't support hundreds of thousands of snow leopards. There just isn't enough food for them there. So that also kind of is consistent with what we're seeing here. These results have kind of confusing, but clear conservation implications. So the main results here are that snow leopards have a small population size. It's always been small, and they have very low genetic diversity, and they've likely always had very low genetic diversity. And so, general conservation biology dogma says that you need high levels of genetic diversity because that is what is going to provide you with adaptive potential to respond to pressures that you encounter. So here in this cartoon, these mice are all genetically diverse. They have different genes, and this is showing that when they encounter this new stressor, in this case, increased temperature, there's two of these mice that have adaptations to deal with this new stressor, and so they survive and can continue on the species. And you can imagine that if all of these mice just had this green gene and this green gene didn't allow them to respond effectively to increased temperature, then they would all die and the species would go extinct. So that's kind of the theory here, that high genetic diversity is just a way to have lots of different tools at your disposal, and be more likely that you'll have something that will allow you to respond successfully to a new challenge. Um, so, so this is why very low genetic diversity in snow leopards could be a problem. That said, there's mounting evidence in other species, as well as now in snow leopards, that very low genetic diversity isn't really a death sentence at all, because the species can survive for hundreds of thousands of years of very low genetic diversity. And case in point here is that snow leopards have survived throughout their whole evolutionary history with a very small population size and very low genetic diversity, and they've managed to survive all of the trials that they've faced throughout that evolutionary process. That said, we are approaching, in this time of the Anthropocene, snow leopards are likely going to be facing lots of new challenges, climate change, habitat change, new exposure to diseases. So, yeah, it's impossible to say what this means, but it could be, it could be dangerous for snow leopards moving forward that they have such low genetic diversity and that they're likely going to be facing so many new challenges in the coming years. Okay, so that was kind of a tangent on step two, filling in this gap of having whole genome sequencing information, and kind of squeezing out all of the insights that we could from that information on snow leopards. And now I'm going to go back to our main goal, which was to develop this SNP panel. So, um, so again, with invasive blood and tissue samples, we can get the whole genome, we can get all 1.5 million SNPs. The first question that we had is from a fecal sample, how many SNPs do we need to get to answer our questions? So we know we're not going to get all 1.5 million SNPs, but do we need 20,000? Do we need 1,000? How many do we need to actually answer the questions that we have? Um, and for those of you who are interested, the method that we're using for our SNP panel is actually different from what many people use, and we've chosen this process in order for the protocol to be able to be deployed in any country. And so what we're using is a multiplex PCR approach, which basically just is, for those of you familiar with PCR, you have your locus of interest that you're trying to amplify. You design primers to sit on either side of that, and you amplify your locus of interest. Here we just do that in multiplex. So we throw in primers for multiple different loci in one pool, and this allows us to amplify out all of the SNPs that we want in one reaction. Um, and so, in order to identify how many SNPs we actually need, our group, led by my colleague Ellie Armstrong, developed this software to feed in the information that we have. So in this case, all 1.5 million SNPs from snow leopards, and tell it the questions that you have, such as individual ID and geographic origin. And it'll identify the SNPs that are going to be most meaningful and most informative for those questions. So in this case, we did that, and it gave us 144 SNPs as being sufficient to answer our questions. So this is just proof that these 144 SNPs do give us a sense of geographic origin of samples. So this is that PCA I showed you earlier where it shows these three genetically distinct groups. This is with all 1.5 million SNPs. Just with the 144 SNPs that we are using in our SNP panel, we can still get a general sense of these three genetically distinct groups. And then this is showing that individual ID can be done with much less than 144 SNPs. So this is a plot where the black points are showing the probability of two different individuals having the exact same SNPs. And the gray points are the probability of two siblings having the exact same SNPs. And what this is showing is that even with only 20 SNPs out of our 144 SNPs, even with only 20, we have essentially a 0% chance of two individuals having the same set of 20 SNPs. So basically, because we've chosen SNPs that are so variable, it's the odds of having two individuals have the same SNPs are essentially zero. So this is just proof that these 144 SNPs are more than enough to answer our questions. So the first thing that we did with the SNP panel was to apply it to scat samples from 5 different individuals at the San Francisco Zoo. And to give you a sense of what the data looks like. So basically here, this is what the raw data would look like, where we have SNP 1, 2, 3, 4 that would go on to 144. Here, this is snow leopard fecal sample A, B, C, D that would go on. And we just look at how similar these are. So here we see feces A is 100% the same. These all have the exact same sequence. And feces A and C is only 15% the same, and so forth. So from this, we can tell that feces A and B are from the same individual because they're 100% the same. And what this plot is telling us is that if we have at least 20 SNPs, you're never going to have, if two samples are the same, they're always going to be from the same individual because the probability of two different individuals having the same SNPs is essentially zero. So this is kind of what the data looks like. What I turn that into for kind of more easy interpretation is a plot that looks like this, where these are pairwise comparisons between all of the fecal samples. So here in this first bin are pairwise comparisons between fecal samples that are 100% the same. And what we know here is that there is some error, so not all of them are 100% the same, but anything that's 95% the same or more are coming from the same individual. So feces A and feces B, if they're 99% the same across all of the SNPs that we amplified, they're the same individual. And then these are all pairwise comparisons between different individuals. So any pairwise comparisons between feces that are less than 90% the same, those two feces are coming from different individuals. And kind of surprisingly, we were actually able to resolve first order relationships from non-first order relationships. So this is again just the zoo samples where there was a nuclear family, so a mother, father, and 2 offspring, and all of the pairwise comparisons between first order relatives, so a feces from a mom and her cub, were more similar to each other than pairwise comparisons between non-related pairs. So once we validated it on the zoo samples, Shaquille came to our lab at Stanford with almost 1,200 fecal samples from across Pakistan. And the first thing that Shaquille did was extract DNA from all of these samples and identify which ones were snow leopards. So this was an insane amount of work, but he identified that 232 of these were from snow leopards. And so we ran the SNP panel on these confirmed snow leopard samples. and did a similar analysis. So this is similar to the histogram that I showed you for the zoo samples, where these are all pairwise comparisons between samples that are coming from the same individual. So any feces that are more than 95% the same, we are saying those are from the same individual. And based on this, we identified that out of those 232 samples, there were 56 unique snow leopard individuals. Um, and this is what that looks like on the map. So you can get a sense of how this can be informative for conservation efforts. So this is just a subset of our data. But individual E here in blue, all of these are fecal samples that were identified as coming from individual E. In purple here are all of the fecal samples identified as coming from individual F. So in this way, you can start getting a sense of the range of an individual. These samples also were collected over multiple years, and I think that we've had like two instances of feces coming from the same individual that were collected up to 4 years apart. So you could see how if you kept on this collection effort over many years, you could start getting assessments of longevity in the species. And most excitingly is that Shaquille has taken this data and applied spatially explicit capture recapture models to it to actually come up with a total estimate for the number of snow leopards in Pakistan. And this work was just accepted and is going to be out in biological conservation very soon. Um, And to circle back to the general geographic assignment, we also see from our fecal samples that we can get a sense of general geography from these samples. So the San Francisco Zoo fecal samples, which are from the captive population, are shown in black stars here, and they are falling in this PCA space where the captive tissue samples are. So if we didn't know where this fecal sample had come from, we could get a sense that it was from this genetic group. And same for the Pakistan fecal samples, which are all shown in pink stars here, which are all falling in this general kind of southern range group. And for fecal samples, this is not so exciting because we collected the fecal samples, so we know where they came from. But this type of insight would be very meaningful for confiscated materials. So if you came across a bunch of snow leopard pelts, if you ran the SNP panel on them, you could identify which general area these pelts were coming from, which you could imagine could be very informative if you're trying to figure out how to curb these types of actions. So where we're going from here, so this snow leopard SNP panel, which hopefully I've highlighted here that it can do a number of things, but is particularly good for individual ID for assessments of range size and population size for an area. I have a protocol written up for this that is publicly available. I'm also happy to share the SNP panel with anybody who's interested in running it. So we've made this as easily accessible as possible. So if anybody is interested in running this in on samples that they have, please let me know. We are also continuing this work and trying to develop a snow leopard SNP panel that's going to be even better for assignment of the geographic origin of species of confiscated materials. And that's because you might have noticed from where our samples came from that we don't really have this cartoon of the genoscape of like the geographic distribution of genetic diversity is only as good as the samples that you have. And again, we don't have samples from a large portion of the range. So we actually don't have this full genoscape. And this is a problem because it means if samples are coming from an area that we don't have reference database for, then we're not going to be able to identify them as coming from there. So if we don't have any reference data from China and we come across a pelt that has come from China, say this green one, we're not going to be able to say where it came from because we have no reference data point. Um, So we are working on this, and we're coming a long way, especially because the Chinese group last year published whole genome sequencing data on all of the samples shown here, which compared to ours, does fill in this gap pretty hugely. That said, we still don't have samples from many samples from India. We don't have any from Nepal or Bhutan. We're working on these. Kazakhstan. Uh, Basically, if anybody has samples that they would like to contribute to the effort, we are still working on filling out this reference database, to make it as useful as possible for this next round of SNP panel. And and then I just wanted to wrap up by saying that all of these methods that I've talked about, again, are applicable to any species of conservation concern. And we do have SNP panels underway for multiple other species. So we're making geographic SNPs for geographic assignment of confiscated materials for plains zebra and African lions. And I already have individual ID SNP panels, more for processing fecal samples for African lions, mountain gorillas, and African wild dogs. So if you're interested in any of these species or in developing a SNP panel for a different species, these protocols are pretty much all the same. Um, and with that, I wanted to highlight that all the snow leopard work that I've talked about, you can read all about in these publications. And we should have time for some questions.
Chris,
Okay.
are the wild population groups identified mixing with each other, or is there a risk that they are becoming isolated groups?
That's a great question. So, that's one thing that we weren't able to exhaustively say with the whole genome sequencing data that we're hoping to be able to address with like our larger data set that's coming down the line. What we found is that they're genetically distinct, but they don't seem isolated. It seems like there's some sort of connectivity, like, but it's hard for us to quantify it. But basically, the measures that we made show that there should be some level of connectivity still, but it's hard to say how much. Um, and uh, yeah, and the isolation is interesting. Yeah, so that's still a point that we need to flesh out more. Because again, yeah, it is important to keep connectivity where there is connectivity. Through those groups, it doesn't look like there's lots of connectivity. So it doesn't, to us, it didn't seem like a main priority that like, oh, there's like main corridors between these areas. It doesn't seem like that's the case. But yeah, we're hoping to get better numbers on that moving forward.
We have a question from Maggie. How old can a fecal sample be and still extract usable DNA?
That's a great question. So Shaquille might be able to answer that even better, but so again, I didn't collect the fecal samples, but in the metadata sheet, from what Shaquille and his team collected, many of them were estimated to be like a month old, and we got them to work. So it's been quite shocking actually. And we're doing this in other areas too, like in Africa. And we're also finding that very old samples work, which is very surprising. And so basically the rule of thumb that I have now for this is that any sample is worth trying. There's no sample that you should look at and just say, oh, it's too old. Like you should always try because, yeah, because I'm just surprised all the time by what works.
Shakil, if you're there, do you have anything to
Yeah.
add?
So we have mostly collected samples less than a month old, and still like some sample were too degraded, and we were unable to extract the DNA properly. 36% of the samples were unable to we were unable to extract DNA from 36% of the samples. So samples more than 2 weeks are likely to compromise DNA extraction. In addition, like it's depend also on the environment, like in northern, the snow leopard range particularly is a cold region, so the fecal sample reserved for a longer time as compared to the other hot desert or other regions.
Thanks, Shakil. Katie, you have a question from Flavia.
Yeah, I can read that. Yeah, what level of technical training and tools are needed for practitioners interested in applying this tool in the field and for contributing? And do you plan to form a database that people can consult, contribute? That's a great question. So, as far as technical training and tools, you do need like a molecular lab, but basically if you are doing so the what the SNP panel is made to replace is microsatellite analyses. And if you can do a microsatellite analysis, you can definitely do a SNP panel analysis. So it's basically just a molecular lab, I guess, set up, nothing so special. What a lot of these other SNP panels for other species are using are very specialized equipment and specialized machines where you have to like, yeah, it's a bit harder. What we've tried to do is make a protocol where all you have to be able to do is run a PCR, and then you can send that PCR product to any sequencing facility and they can sequence it for you. So yeah, and then as far as a database that people can consult, well, all of this data is publicly available. We are hoping at some point, this is the pie in the sky thing where it would be great to have what we've been saying is like a 23andMe for wildlife, where you can have like an interactive database type thing where you can add your samples and it can tell you like where they group with and tell you about your samples. That's still like the thought, but I'm not a coder, so I don't know. I think it's going to be a lot of work to get a website like that up and running, but that is kind of the ultimate goal, so that you can just run your samples and then add them to a publicly available database and figure out how yours compare to everybody else's. And I'll add that if you are interested in running it, I'm happy to talk with you as far as like what tools or equipment you might need or who to collaborate with to be able to do it.
Katie, you had mentioned that you weren't able to get samples from certain areas, I mean, certain of the range countries, and you said that you were trying to deal with that. What are your plans about that?
Yeah, that's a great question. So I was going to go back. So these samples from across China. So China's been the hardest one to deal with because I think there's all sorts of like regulations within the country about sharing data and stuff. So this has been a huge windfall that all of this data is already publicly available. So these are already samples that are essentially in hand. We are still working with colleagues in Nepal to try and fill in some of the gaps there. Those are the main ones on the radar. We do have samples also in Kazakhstan that we're trying to get our hands on. What's more problematic is just the regulations of moving samples and processing them. So we're trying to figure out how to process them in country, which, for example, in Nepal right now is proving really hard for this mere fact that the reagents that you need to process the sample are just hard to get to Nepal because they're made in the US and they need to be kept cold. And so yeah, the collaborators are amazing and very happy to help. It's more just the practical issues of getting things places that's proven difficult. But I think by the end of this year, we're hoping to add a handful more samples. And actually, I should highlight too that from Shaquille's effort, we already have like four more samples from Pakistan that's going to be added because currently in our current analysis, we just have one sample. So we're going to have a bunch more there. So yeah, I'm excited about the next round of analyses.
And there's also a lot of snow leopard work happening in India. Why are samples from there so rare to come across?
That's a great question. So the one sample that we do have from India is from Uma's lab. And I think there's additional samples. India also samples have to be processed in country. The samples can't leave the country, which is very easy in India because there's lots of genomics facilities in India for them to process them in country. So we will have some more samples coming from India, but I think Uma's working on some stuff too. So I don't know exactly, but I do think most of them are from this area of Himachal Pradesh, and there's still none from like Sikkim or further east, which yeah, I don't know what the deal is there. If people maybe just yeah aren't studying them there so much or what. Is it possible to use samples from other teams collected in the field to analyze? Yeah, for sure. Yeah, so all of these samples that we've used are from other people's collecting them in the field. This is kind of hot off the press information that I'll just leave out there that it actually might be possible to get enough information from fresh fecal samples to kind of fill in some of these gaps too. So so just to say that if you come across fresh fecal samples, that could almost serve the purpose of a we're finding that that can almost yield as much DNA as a tissue sample. And but as far as what, okay, and then you asked what's needed for them to be viable for analysis. Any blood or tissue sample that's been kept like reasonably well. There are different preservation methods that are better, but again, anything is worth trying, and I'm happy to like look at a case by case basis. But anything is worth trying. Um, and are there any major limitations from this analysis for conservation practice? Um, yes, okay, that's what I was gonna say. Risk is, yeah, yeah, there are. And luckily, because this has been such a collaboration with all these groups, like when we share this information with our colleagues at Panthera and Snow Leopard Trust and stuff, like I make sure to tell them like take this with a grain of salt. But like those graphs that I showed of like historic snow leopard population size, like those need to be interpreted with a huge grain of salt. Those are like order of magnitude numbers for snow leopards. So like we can say realistic, like for sure there weren't hundreds of thousands of snow leopards, but you know, it's like an order of magnitude that like there were like 10,000, 20,000, 30,000. Like so those can't be overinterpreted. Um because they kind of wanted to interpret those heavily for like current population sizes too, but that really can't be done. And then these connectivity things too, I also hesitate to say too much uh, because it's just hard, and people do want clear answers of like how many snow leopards are moving between Mongolia and Kyrgyzstan every year. Like we can't say anything like that. We can just kind of get a gist of like, oh, they look different, but not so different that they're like completely isolated. So yeah, so there is you all who kind of are more on the ground, like doing the conservation work, would have a better sense of how things could be misinterpreted. But I try and be pretty realistic about what exactly can be said and what can't be said with certain information.
And Katie, is there a time tenure on the samples to be viable? Like, is there a point where you just give up on trying to get any more information from them, or does that really happen?
Do you mean fecal samples or
Yeah.
So Shaquille had more insight that like he thinks that once a sample is of a certain age, it's not worth pursuing, and that's only um, so we're talking about the age at which it's out in the field. But like Shaquille's samples are multiple years old now, and they're still sitting in our lab. So like once you collect and preserve them, I think they can stay viable forever. But yeah, but the fecal samples, Shaquille is saying that that they might not be worth collecting after 2 or so weeks. But like I said, my work in Africa is showing me that stuff that I would never bet money on working does seem to work. So I think the success rate declines a lot. But because snow leopard samples are so rare to come by, it's I my motto is that it's worth collecting everything because even if 1 out of 10 works, that's still, yeah, worth it.
Thank you so much, Katie. Do we have any more questions?
Oh yeah, if people with no training come across a sample, how could they collect it and a way to use? Um, this is a great question that I have a long answer for. Um, Fecal samples, we've been collecting in multiple different ways. The way that Shaquille collected all of his samples that worked really well is just collecting it and putting it in a tube with silica beads, and drying it out, basically. Um, the best way if you aren't prepared to collect a sample, if you have access to a freezer, that's the best way is put it in a bag, label it and throw it in the freezer and then just kind of deal with it later. If you can get it into a freezer, then it's kind of good and you can figure out what to do with it later. Now, if we're prepared, we're using more of a buffer called a Zymo DNA RNA shield. And those are like 2 ml tubes, which are a lot smaller than the tubes that Shaquille was using, that you can just carry around in your pocket basically and do like a little swab from the outside of the feces, or just take a little pinch from the outside of the feces and put it in this 2 ml tube and shake it up, and then it can stay at room temperature for weeks or months. Um, so basically, there's lots of ways. It's better if you're equipped for it, but if you think you're going to be encountering some, I'm happy to talk to you and tell you what to put in your bag to be able to collect them.
Katie, you have a question from Shiv Kumar as well?
Oh, awesome. Okay, I'm working in a frontline staff in the Himachal Pradesh Forest Department. How could we contribute? Um, Yeah, so I mean, fecal samples or tissue samples would be amazing. So, tissue samples, again, for the reasons I described, are the best for like this reference database. Tissue samples, if you come across like a carcass or even like hair from like scratch marks or something, those can be collected in lots of different ways. I'm happy to give you more information, but like ethanol is good, or this Zymo DNA RNA shield. And then fecal samples too are great to collect. Um, but yeah, I feel like I'm happy to talk more specifically, because that would be an area that would be great to get more samples from.
Thank you so much, Katie. I think we're out of time now, but I put Katie's email in the chat. So if anyone would like to get in touch with Katie, thank you, Katie, for sharing that. And please do email Katie directly. Thank you, Shakil, also for facilitating this wonderful presentation. The wealth of information in this small little presentation was so much. I think we could all have questions for you forever.
Well, please email me. Yeah, I'm happy to talk.
Yeah, and if anyone would like to get in touch with Shakil as well, you could email me and I can send you the email address. If that's okay, Shakil?
Yes, okay, fine.
Okay, so great. That was wonderful. Thank you so much. I mean, I feel we really learned a lot. Very informative.
Awesome. Great. Thank you all
We
so
look
much.
forward to, yeah, look forward to hosting Katie and Shakil again after the papers are published. And when we have more information, we look forward to hearing from you.
Awesome. Thank you all so much for your interest. Thanks, Shaquille. Thanks,
Thank you,
Rocky.
thank you.
Thank you.